Cool. So my name's Carl St. John. I'm a PhD from Cornell University, and today I'll be talking about genetic and phenotypic convergence on an extreme life history variant in the white sucker, which is a fish that we find all over North America. I'll be talking about it in the Adirondacks. I'd first like to acknowledge all the wonderful people who've worked on this project. They come from all across New York State, and it's been really fun to travel around and sort of unite people around the cause of the suckers. I'd also like to acknowledge that much of this work took place at Cornell University, which is located on the traditional homelands of the Gayakono Nation. And then much of the field work took place on the lands of the Mohawk Nation. Both of those nations are members of the Haudenosaunee Confederacy, which outdates both Cornell University and this country. And we acknowledge the history of Gayakono and Mohawk dispossession and their presence on the waters and lands that I've done this research on. So I think a theme that many folks have been interested at least in the talks I've seen so far, is the repeatability and predictability of evolution. So if you end up on one phenotype, how many different ways are there to get to that phenotype? And cases of repeated evolution among closely related demes, a la adaptive radiations or similar scenarios, are a very common way of studying this because in theory you're starting from the same evolutionary material and ending up with the same phenotypes independently. The Adirondack region of New York State is a great place to do that because it contains over 3,000 different lakes and ponds. Within some of those ponds live white suckers and their extreme life history variant, summer suckers, which I'll be talking about today. White suckers, so these we're talking about catastamids for any fish people out there in the crowd. White suckers live all across North America, all the way from the high Arctic down to New Mexico and Georgia, whereas summer suckers, their ecotype, just lives in the Adirondack region of New York State in that little purple bubble. Summer suckers are very small, so they mature at less than 200 millimeters, whereas white suckers will mature at much larger sizes. If you need a device to remember that, you can think small summer suckers, and then the best I could come up with was a whole lot of sucker for white suckers. So keep that straight if you can. White suckers so there's also spawning time divergence. So white suckers will spawn early in the year in the Adirondacks, around early to mid-May, and summer suckers will spawn later in the year, early summer all the way into July, but note that considerable overlap. White suckers are spawning before leaf out, when things like snowmelt is primarily driving the hydrology of the system, whereas summer suckers are spawning in late summer, where like thunderstorms like this one are driving most of the hydrology in the system. So just a quick glance at population structure in the Adirondacks. If you sequence individual white and summer suckers from the same lake, you'll note that those individuals will group by lake rather than by ecotype. So this tells us that these are sort of independent evolutions or likely independent evolutions of that summer sucker, that small life history variant. We are going to study two of those lakes in the Adirondacks, Elk Lake and Deer Pond. You'll notice that those lakes group by geography rather than by ecotype. And they're located in totally different drainages in the Adirondacks, so they don't share much evolutionary history. So we're going to try and combine phenotypes with genotypes and figure out if summer suckers have evolved independently more than once, or if they've evolved through sort of parallel pathways, both genetically and phenotypically. To do that, suckers will live in lakes predominantly, and they'll migrate into tributaries to spawn, and we can intercept them on their spawning migrations to get an idea of the time of year that they're spawning. So we catch them in a trap net on their way up to spawn in their tributaries. We sampled at regular intervals over the spawning period of both ecotypes, and we collected fin clips for genetic data, lengths, weights. We vouchered some specimens to get other phenotypes, and then we sexed individuals. And again, we did this in two sort of replicate systems, Elk Lake and Deer Pond. Our sequencing methods were low coverage whole genome sequencing. This just means that we're going to be working with genotype likelihoods rather than hard-called genotypes. And so our analyses take into consideration the uncertainty we have over the, as to the genotype of any given individual. Our first stab at looking at genetic variation within lakes is, of course, a PCA. In Elk Lake, on your left here, you'll see a PCA of all individuals caught in Elk Lake, and it's colored by phenotype. So on the top is spawning time, on the bottom is body size, and you'll see this sort of correlation between genetic variation explained by PC1 and both spawning time and body size. In Deer Pond, on your right, you'll see maybe a slight relationship between spawning time and genetic variation explained by PC2. But no such relationship for body size in Deer Pond. So we're already sort of thinking more about convergence than parallelism at this point. Just to remind you, we're talking about fish, not dots. So yeah, we see this sort of continuous variation between the large white sucker ecotype and the small summer sucker ecotype. But PCAs only give us this sort of bird's eye view of the genome, and we know that genetic variation can vary heterogeneously across the genome, and that can tell us about how evolution and selection takes place at different points in the genome. So we use genome scans for that. So as you may have picked up, there's sort of this continuous variation between ecotypes here. And so instead of showing you a FST genome scan, which is pretty common, I'm going to use GWA, which can process continuous data. That's what you see here. So this is a GWA for spawning time on the top and body length in the bottom in Elk Lake. You'll find that we identified some outliers. We used the local score method if anyone's interested in talking about outlier detection methodology. And we found sort of this polygenic trait architecture throughout most of the genome with a locus of seemingly large effect on chromosome 49 here. I don't recommend working on organisms with 49 chromosomes if you can help it. So we did this for Elk Lake, pretty interesting results. We also did this for Deer Pond. And in Deer Pond, find a different story. We find one locus associated with spawning time in Deer Pond, and this locus seems to have less effect on spawning time, or at least not as strong of association. And then we find these three outlier SNPs associated with body length in Deer Pond. So you notice, there's localization to different genomic regions in these repeated evolutions of this life history variant. So pretty strong evidence that there's convergent evolution on the same ecotype. We can dive a little bit deeper into those outlier regions by looking at how diversity varies over those candidate regions. So here's a zoomed in view of the chromosome 49 outlier region. You'll notice that both nucleotide diversity and Tajima's D are locally depressed across this region. This suggests to us that maybe non-neutral selection is occurring here. I'll also note that the effect size of these genotypes seems to be fairly strong. So on the bottom left here, you're looking at allele dosage. This is like an estimate of the genotype across a region of the genome. And then on the x-axis, you're looking at a phenotype. So for spawning time, you see this relationship between genotype. So some white sucker individuals always spawn early versus summer sucker individuals can spawn over this larger range of the spawning season. Then we see more of an additive relationship between genotype and phenotype for body length. If anyone's interested, we did the gene fishing trip, and I can come up with any number of reasons why we found these genes, but not for this talk. So we did the same thing for Deer Pond, and we found sort of more fuzzy results. So we do find locally depressed Tajima's D, selecting non-neutral evolution, or suggesting non-neutral evolution. We find a similar relationship between genotypes and spawning time in deer pond, but for body size on the lower right here, we don't really find any relationship between genotypes and body size. And so that sort of squares with our PCA and GWA results, where in deer pond, this ecotype or this body size trait seems to be more plastic. Again, fun fishing trip. This was a really fun one, so ask me a question about it if you want some grasping. Okay, so what does this tell us about the repeatability and predictability of evolution? Well, at least for life history traits and in suckers in the Adirondacks, it looks like cases of convergence across the board, both in phenotype and in genotype. So for Elk Lake, body size has this genetic component, but body size seems to be more plastic in deer pond. Spawning time has a genetic component in both systems, but different places in the genome. And then in Elk Lake, we see this polygenic / locus of large effect trait architecture, and in deer pond, we just see this single locus of small effect. Cool. If you're not jazzed about suckers yet, note that lots of cool things eat them. This is usually like if I'm presenting to a management crowd and they're like, summer suckers. Lots of charismatic species depend on these fish, both for nutrient cycling and for prey. And with that, maybe some of you study these individuals that I collected in my nets that were not suckers, and I'll take questions.
That popped up in your analysis. So I study salmon life history variation.
Yeah.
And NR5A2 is popping up as a really important gene in salmon.
Oh, that's super cool.
Maturity,
yeah.
So size of maturity variation too. So maybe we should talk.
We should definitely talk. Yeah. And yeah, for sex, I know in salmon there is sometimes like this sex-linked trait that determines migration variance. In this case, across the board, males run before females. But you'll see I broke up the spawning time into males and females, and it's sort of like the effect is shifted to the same magnitude between males and females, but it doesn't play out differently between them. Yeah, good question. Anyone else before the Grim Reaper bell tolls? All right, let's get the next person up here.
So hi everyone, my name is Pavel. I'm a postdoc in Nina Turkleton's lab. I'm in the same lab as Carl, and Carl just defended his PhD 2 days ago. So when you see his golden curls later today or during the conference, congratulate him. So we're going to be covering a lot of ground in this talk. We're going to be talking about some fish stuff, some genetic variation, some inversions, and a relatively new sequencing methodology to top it all off. So let's just kick that off. So we're in sort of like a pop gen, right? It's a familiar concept to most of you, but just to make sure that I don't lose anybody, typically you have some sort of species or entity in space, in time, and we used to think that it all used to be one genetic entity. We now find out that more often than not, it's actually multiple smaller groups of something interacting in space and time. And so now that there's groups of stuff, we have to consider how they're interacting or if they're interacting. And for the sake of this talk, when I say interacting, I really just mean like gene flow, sex, scandalous things. So there could be all sorts of things happening between these groups, right? There could be barriers between them like that. I don't know if you could see it, not super contrasty, like the yellow barrier between the top two groups. Could just be distance, which makes it hard to travel between different groups. And over time, these sorts of things can result in differentiation with the things of like selection and drift. So hopefully those aren't new concepts. And in like terrestrial species, there's a lot of conspicuous things that can be causing those things, right? Like something from California versus something in New York. It's often quite a big distance to travel if you're a snail. Probably not going to. A mountain range can separate mice. But when you're talking about, say, marine fishes, the marine system is kind of like just a homogeneous thing. And so we're not really sure what happens between these groups or what these groups look like. And it's even complicated further in the marine system that marine fishes tend to move and coexist with each other in space and time, even though they might not be genetically interacting with each other. And so, enter the frustrating world of marine population genetics of highly migratory fishes. So let's talk about one of those. The study system that I will be discussing today is the American shad. It is the largest of the herrings. It's in the Clupeiformes family, so similar to other sardines and anchovies, it's a planktivore. This one is anadromous, which means that it moves between marine and freshwater systems. And it's not doing super well in the eastern US, and we'll discuss why. This is its native range in the eastern US in purple, and so obviously it has a marine area, it has a freshwater area, and for reference, we're about here, so we are currently in shad country, although the shad run ended, so you're not really going to find them in these waters. But shad is actually a really important fish historically in the United States, which I know we're in Montreal, so that doesn't exactly have the same traction, but it was a really critical source of nutrition in early colonialized America and also for indigenous communities. When shad do their annual runs up rivers, that is a huge bottleneck to just catch them in nets, and they're quite nutritious, although people don't so much eat them now as often. So here's like a historical photo of people gathering around a net as the shad runs are coming in. Here's an even older sepia photo of something similar. This fish was critical for like early wars in the colonialized US as a source of nutrition, and it's even been called a founding fish with a book written about it because it's actually quite significant. You would think that something that important would want to be preserved or conserved, and it's not. This is a map. If you've never seen it, this is a map of the dams in the eastern US. The blue are the dams. So there are a lot. And shad, of course, because they go up rivers to go to their breeding areas, suddenly don't have access to those breeding areas. And they're being exploited as they're coming up rivers. And then you add the Industrial Revolution to that and pollution, and you have crashing stocks, right? And like, that's not surprising. This is just data from one river, but they pretty much all look like this. So in the title, I say it's imperiled. Clearly, it's not doing well. And there have been efforts to restore it, which we'll discuss. But when you want to do a project on something like this, you're like, okay, here is a place where it exists. Let's sample it there and do some genetics on it. If you don't work with marine fishes or highly migratory ones, the first thing you will often find out is that the statistical values you get for these things are super low compared to other systems. So you have to sample very carefully in these systems. So if we were to just sample them when they're in their marine phase, where they're physically admixing, you would just catch a whole mishmash, and then you would probably get a lot of noise, and it'd be difficult to figure things out because you're more than likely sampling many populations without any like actual extra distinction in the data to suss it out. However, shad are anadromous, and so we could just sample them when they return to their rivers. It's a very exploitable life history characteristic. So we did. We caught them. And this is work done by Isabel Kim. So on the right, you have a PCA of the allele dosages, which this was low coverage whole genome data. So again, as Carl had mentioned, if you'd seen his talk, you're not typically working with genotypes, but you're working with genotype likelihoods, as it's unlikely to call very confident genotypes at like 0.5 to 3x coverage. But you see that there is clustering in these rivers, which are color-coded with the rivers on the left. So you have Miramichi and Annapolis, two rivers in Canada, cluster separately, and then everything seems to have this fine structure to it. But when you do an admixture analysis, which is in the center here, every individual from every river is represented as a horizontal bar. The ones at the very top, which are the two Canadian rivers, seem to be population 1, and then the ones at the very bottom in Florida and South Carolina are population 2, and seemingly everything in the center is some sort of admixture, right? You would think that with the combination of bars it would be. And you're like, wow, that makes sense, right? Canada, Florida, and then everything in between is just a gradient. But shad were stocked. As I said, shad weren't doing so hot because we wrecked them, frankly. So we're like, okay, let's catch them, let's grow them, and then or move them in between rivers. So Dan Hasselman, when he was doing his PhD, did the insane task of compiling all possible records of shad ever being stocked. And I can't imagine how much work that was, but he gave me the data and asked me to plot it. So this is what it looks like. And if this looks busy and overwhelming, it's because it is. So if you've never seen a circle plot, this is the worst example of how to interpret one. But every arc is a river, and the bands, the width of them indicates how many fish relative to other ones, and the directionality of the arcs suggest from what river into what river. And so I could see that you guys are concerned, some of you are grimacing because, yeah, it's insane. So shad were stocked a lot. I should mention that the colors correspond to the decade in which the stocking happened. So it's a lot, and the Susquehanna River got the most of them. So, you know, we have this like cool admixture, but we don't really know if that's natural. It could just be human induced, right? So another thing was that the stocking was actually unsuccessful. It turns out shad don't want to get better, right? What if they tried harder? So shad don't do well with the dams, right? The dams are still there, and so you could stock them all you want, and they're not really accessing any breeding areas, and even though there's fish passages and ladders and elevators, they're not using them very effectively. So this isn't a fisheries talk, but if it was, big thumbs down, you know, consistently. But, you know, we're talking about what the populations look like, and so there's the question of, you know, can we suss things out better, and will we need more powerful tools to do it? I had mentioned that this data was whole genome and low coverage, but it was also linked read data, which was all sort of done in one go, and I'll explain that in a second. So if you're not familiar with sequencing stuff, here is a diploid chromosome. One chromatid is in blue, one is in black. Typically, when you want to sequence on an Illumina machine, you have to fragment things to about 300 to 500 base pairs so that you're not just wasting money. When you do that with enzymes or any other fragmentation, you dissociate or lose the information of which fragments were actually together on the same exact chromatid in the same nucleus, which, you know, could be useful. And with RAD, if you're doing enzymes, same deal. However, linked read sequencing, what happens is at the fragmentation step, it also adds a barcode right then and there to all of the fragmented DNA that came from the same original strand of DNA. And if you think about that for a second, that's every strand of every fragment that came from one single molecule of DNA from one single nucleus would get tagged with the same barcode, which means theoretically downstream, you could just find that barcode and associate every fragment with the same chromatid. If you're not very bioinformatically inclined, that has like huge downstream implications of how you can use those data. Our lab is a big proponent of haplotagging. It's a relatively recent method of linked read sequencing. There's only like 3, I think, existing methods. That's the third one. Of the 3 current options, it's the only one that is non-commercialized, or I should say not for profit. And we also process these data with an in-house software that we built called Harpy. Not Python, we didn't build Python. So one of the things that we could do with this linked read data is go from these genotype likelihoods, which we still get with the same exact low coverage information, and actually impute genotypes with it so that we can convert to using actual genotypic data instead of likelihoods. So here is just a subset of those shad individuals. Every horizontal bar is all of the SNPs that that individual has, and the dark gray is things that we could not impute, and the colored stuff is the genotypes that we were able to call with the imputation with this extra information that we have already had with it. So that unlocks a whole new set of statistics that we could be doing. In addition to that, if you've ever had to phase SNPs into haplotypes, you can do that too. So these 2 bar plots, not the shad data, they're fly stuff. But if you look at the top bar, I don't know if you could see from the back, but the y-axis goes up to 60,000. That's the count of haplotypes. And then the bottom one goes up to 2,000. And so these are the same exact fly data, and it's phasing without the linked read information on top and phasing with the linked read information on the bottom. And so we get substantially fewer but much, much larger haplotypes. Because there was an extreme right tail that went up to 7 megabases, I just collapsed everything that was above 25 kb into a single column. And so phasing is cool and useful and lovely, but we also have been able to call structural variants with these same low coverage short read data. And so I'm sorry I don't have like big honkin' inversions to show you, but here are like 3 circle plots just for summary's sake. Each wedge is a different chromosome of the shad genome, and then because we're doing multiple populations, I arranged this one such that every ring is a different population. And you can find inversions, duplications, and deletions. So if we were to say zoom in on a single inversion that we were able to identify and yank out all of the SNPs in that interval, do we find anything interesting about it? And for the inversion that I chose for this presentation, no. Unfortunately not. Although we have other evidence in other shad populations that there's actually some very interesting patterns that emerge and structure that occurs at these larger inversions. And so, the short version is we're still working on it. This is kind of cutting, I hate using that phrase, but this is kind of cutting edge stuff, and I'm the one cutting the edge with it. So, I was only able to present so much today for it. So we're working on it, and it's a pretty big collaborative effort, and I'd like to highlight the people that are working on it. That includes Nina Turkleton, who's also giving a talk later in this conference, and I suggest you go see it, as well as Oswald, who's giving another shad talk on Tuesday. Do recommend you go see those. But yeah, this is part of a large collaborative effort to sort of push the idea that linked read sequencing can just be used in tandem with what you're already doing to get more out of what you're doing. So thank you for your time. I have, I think, 1.5 minutes of questions if anyone's got anything. Yes?
So how does the information of what chromatin or read comes from help with imputing gene effects?
So the imputation method that we've been using relies on the barcoded information because it haplotype phases internally, and it's helpful for the phasing process specifically. Yeah. So it reduces the uncertainty of like which SNPs would have been associated with each other because you kind of know whether or not they came from the same molecule. Yes?
I was just wondering, what did you use to call structural variants?
Ooh, that's a good question. So part of the tricky stuff of linked read analyses right now is that the software space isn't huge. And so there's a software that's called Leviathan, which is the coolest name ever. And that one doesn't work as well as this other one called Neighbor, N-A-I-B-R, which Leviathan's easier to use, but Neighbor requires you to phase your SNPs first to phase your BAMs to call structural variants. And so if that sounds confusing and difficult, that's why we created the software to do it for you. But yeah, you could talk to me after about it if you want more details. Yeah. Anyone else? We'll talk after. Thank you for your time.
So yeah, hi everyone. My name is Sozos Michaelides. I'm a postdoc here in Montreal, Concordia University, and I'm excited to present you this work I've been doing for almost 3 years on ecoevolutionary processes that fuel intraspecific variation of lake trout in Great Bear Lake. You might have heard earlier today about how important maybe is intraspecific variation, and it's increasingly being brought up in publications how studying genetic, genomic, and phenotypic variation within and among populations is important to understand ecological processes, but also to conserve this diversity as an important component of this major global biodiversity crisis that we go. We can also think the intraspecific variation along the speciation continuum as well. We might think, for example, sympatric morphs, like those that we see mostly in, like, say, in salmonids, represent an interesting idea of how we can study them to uncover the mechanisms behind that giving rise to such variation. Multiple examples that maybe it's just plasticity, that we don't see any genomic regions associated with, for example, piscivorous and plastivorous morphs. Or by increasing differentiation, maybe we pick up structural variants or some large effect loci. But also it might be the widespread that might lead eventually maybe in some speciation events. So one of those salmon species that I'm interested in and I'm presenting today is lake trout. It's a large salmonid species that is found, as you see in the map, widespread in North America. And it occupies mostly post-glacial lakes, so they are evolutionarily young systems, of 8 to 40,000 years old. You might have seen this picture before. This is intraspecific variation of these morphs that are found widespread in multiple Great Lakes here in North America. Most of this diversification has been associated with the depth partitioning. All of smaller lakes have maybe 2 morphs, larger lakes have up to 4 morphs. So it's interesting to understand here this variation within the species and what this means for biodiversity. I'm going to draw your attention here for Great Bear Lake, the northern Great Lake here in Canada, which is the 9th largest lake in the world. And you can see here maybe has like 5 basins or 5 arms of the lake. I put there in red the indigenous community that resides in Deline. And I should say that lake trout, those other salmon species, are important for indigenous subsistence fisheries in the region. Previous work on lake trout has identified 4 morphs, as you can see here on the left, and they were mostly found in the shallow water. So most of the past work in that system was focusing on the shallow water. So for us going in now, we want to see what's the extent of intraspecific variation. Can we find more variation maybe in deeper parts of the lake? Maybe also what are the mechanisms that generate and maintain this variation? And can we maybe try to use genetics to find the genomic basis of such differentiation? So to do so, we extend the sampling from past years to include many regions, especially in vertical throughout the lake, almost 400 fish. And as you might have heard earlier today, we use low coverage whole genome sequencing. We follow recommendations. We use ANCs most of the analysis. The full data set was 3 million SNPs, and then they reduced around 60,000 SNPs. We also analyze photographs, and we focus on measuring some specific traits associated mostly with trophic ecology and locomotion. Okay, I'm going to jump into pop structure results. So results indicate that at least 8 genetically different lake trout groups in Great Bear Lake. Here you see an NGS at mix or structure plot. I'm only showing you here the non-admixed individuals. See how all these vertically individuals are clustered together. But we also found substantial admixture in this system as well. Focusing on the non-admixed individuals, the PCA looks like this. Again, the 8 groups. But we start now seeing the pattern that PC1 mostly explains body size, and then PC2 explains roughly the water depth, and I'll come back to this in a minute. Another way to visualize the relationship between these 8 groups is using a phylogenomic tree from IQ3. And we can also start seeing, again, maybe the relationship is of some of these groups. For example, you might see, can you see here? Well, yeah, I'll come back to that as well. So genome-wide FST values range from 0.016 between the group 4 and group 5, but all go up to 0.6 between group 1 and group 6. So there's quite a range of FST between these 8 groups. So now, digging more into the axis of divergence. As I said before, there's variation in the water depth. And I plot here on the box plots below the variation that you see in the water depth, of these 8 groups. We have, the water depth, I can't really read this. So we have roughly 4 groups are found in the shallow water, as been shown in the past, but also we found groups that are predominantly were captured in mid to deeper water levels. There's also a significant variation in the proportion of lake trout found throughout the lake. Here I'm showing you in these pie charts the proportion of non-admixed individuals found in these regions. So we can start seeing that some particular basins or arms of the lake, you find more of individuals of one particular group. There's also phenotypic variation as well. Here I'm showing you some examples of 4 traits: body size, maxilla length, jaw length, and pectoral fin length. So there is variation in the phenotypic traits that we measure for these fish. Now, putting all this together, the spatial variation, the vertical variation along the water column, and the phenotypic variation, let's explain a little bit more these groups. So we have two giant lake trout. One is found in the shallow water, and one is found in the deeper water, here indicating orange and yellow. They live long, up to 50 years old. The third group here is a shallow water group that is found exclusively in McVicker arm of the lake. I'm not sure if you can see this. Then we move on to two deep water, or fish that occupy largely deep water regions, A and B. They vary a little bit on the vertical distribution, but there's no map indication of the spatial distribution of those. Then we have three more groups that are found mostly in specific arms of the lake. We have one, this purple one found in McTavish, one midwater one that is found in Smith, and another one found in this arm. The last one is also differentiated with the pectoral fin length, and the locals call that the butterfly one or insectivore one. So now going into understanding, or trying to understand, I guess, the genomic basis of divergence. Do we find any evidence of selection? So what I'm showing you this figure again about where do we find the proportion of non-admixed individuals. But it's also interesting to look at the admixed individuals here. So the map below, each dot is an individual. It might be hard to look at the pie charts, but they show you a proportion of membership to all these 8 groups. And if you put on them on the 2 axes, the proportion of you have from group A versus group E, and the expectation along that dotted line that I have here, well, if you're a hybrid, you might have 50%, and if you're F1 or backcross, you might end up along that straight line. So I did that with all the pairwise comparisons here, and of course, we found mixed results in the sense that we have groups like the 2 giants, for example, that we find hybrids along that line. Similarly, with the second deep water group and the shallow water group that's found in McVicker. And as you can see from that arm of the lake, there you see most of the individuals are found there because, well, that shallow water morph is found predominantly there, so there's likelihood of them being hybridizing. But we also found examples that we couldn't find hybrids in the system. So between the giants, the deep water giants, and the deep water group, there's no hybrids between them. Similarly, with the other deep water morph and the shallow water morph found in McTavish, and also the deep water giant and the other group in McTavish as well. Now, looking at the genome-wide FST by plotting them along the genome, here, lake trout has 42 chromosomes, and I'm showing you all the 3 comparisons that we saw before that hybridization was not common. And as you can start seeing that, well, there's some peaks of differentiation between them. I'm highlighting first chromosome 1. This peak along the chromosome 1, if we zoom in, we can see this peak towards the end of the chromosome. We also pick up some interesting variants along chromosome 14, 19, and 30, and looking at the linkage equilibrium and see, well, maybe there's some blocks there of islands of divergence. Maybe this might indicate, well, there might be some chromosomal divergence happening in the system, explaining the differences that we see, maybe the lack of hybridization. Now, returning to the phenotypic data that we collected, we did a GWAS analysis on some of the traits I'm going to show you now. First, body size. As you see, remember there were 2 giant groups, and then the remaining of the groups were mid-sized and smaller-sized fish. And we found strong evidence, again, on chromosome 1. Similarly, as we've seen before in the analysis, that there's something on chromosome 1 that maybe differentiates the larger fish. I haven't fully annotated the region there, but previous research has shown that chromosome 1 contains growth-related traits. I briefly look at one of those outliers, and it seems like there might be genes related to lipid metabolism. Another trait that may differentiate the deep water, or maybe the morphs that they occupy deeper waters, and the maxilla length, so head and mouth shape. When you did GWAS, I didn't find any strong outliers, but there's something in there that might indicate further investigation. The third trait I'm showing you today is the pectoral fin length that differentiates this large long fin group of the eighth, the butterfly one. There's some large outliers there that's showing on the plot. Again, I don't have an annotation for those ones. It might be outliers that I need to look into more detail and see if that's significant or not. Okay, so to summarize what we've learned from analyzing and extending the sampling from this system, there's at least 8 genetically different groups. Previously, the research has focused on the shallow water and identified morphs based on their morphology. And now with the genetics, we were able to find at least 8 genetically different groups that vary in the spatial and vertical variation, phenotypic variation on body size and fin length. And what are maybe the mechanisms that we find? Well, there might be partial reproductive isolation, maybe from the non-random admixture that we found. Maybe the timing or the location of spawning might be different between these groups. There might be also evidence of selection through those genome-wide scans, some peaks of high FST outliers that we found. But there may be also indications that maybe we have some structure variants there with the chromosomal inversions. So all this in remarkable diversity, intraspecific level is important for Great Bear Lake, and it's important to conserve this diversity, especially for the local community and for future generations. And with that, I want to thank all the collaborators and the welcoming that I had last year that I went to the community to present these results, and of course the funding, and then everyone here attending. So thank you. Yeah.
Thanks for that talk. I was wondering if the indigenous community had any traditions associated with the different morphs that was already known, and if you were able to work with them to describe that or relate their knowledge to your findings?
Yeah, I think that's a great question. So we are part of the Fishes project. We have a social team that actually goes there to the community, and they are working with them to interweave traditional ecological knowledge with the genetics. My role last year is I presented the results, and I talked to the community, what do you think about having this biodiversity? The community have their own ways of seeing these fish through their traditions. They recognize 5 different types, but of course they don't have the genetic tools to understand these things. But it's interesting to interweave both perspectives here for them as well. Thank you.
Well, my name is Maria. I'm postdoc from the University of Helsinki, and today I will talk to you about the life history, variation, and genetic diversity and structure in a population of Salmosalar from the northernmost distribution. I will briefly introduce Atlantic salmon, that is an heteroporous fish with the most varied life history strategy worldwide. These strategies varied based on the time they spent in freshwater and the marine phase. Typically, they spend about 2 to 4 years in freshwater. Then they have a long marine feeding migration to recondition for about 1 to 3 years, and then they have a subsequent homing back to their natal river to spawn. Well, variation is added to this life history for those individuals that are able to survive and have another event of spawning. Well, the abundance of Atlantic salmon have faced decline or already been extirpated over the past 2 centuries, mainly due to human-related activities. But there is still some natural spawning system in the northernmost distribution. One of these is this transboundary river between Norway and Finland, where the release of rare fish and eggs is banned. And however, this system have shown a decreasing trend in the salmon run size from 2006 and 2022. So these features offer an excellent opportunity to monitor the status of a salmon river in its near natural conditions. So, we study the life history strategy variation based in 22,000 fish scale collection from 1975 to 2022. For determine the life history strategy of each individual, we use scale growth rings. Then we evaluate the trend of the proportion across the time of the most common strategies for this particular river that correspond to 1 to 3 sea winters, and the repeat spawners, because are important for the population, using a generalized linear model. Then we evaluate juvenile samples from 8 locations along the river using a 60K array SNPs, and we estimate the genetic diversity, the structure, and we estimate the contemporary effective population size. Besides, to try to explain the structure that we found in this system, we compare with another system that we have access, that is Teno River. So, for this system, we found a wide life history diversity with combination of small ages, which goes from 1 to 7 years, and from 1 to 4 years for maiden salmons, and several forms of repeat spawners. And all of them comprise about 69 unique strategies of life history in the study period. The most diverse group that we found was the repeat spawners, that although you can see that they have a low proportion that is only 4%, they contribute to about 61% of the variation for this population. And well, the oldest salmon that we found in this data was a 13 years old female. Then we rank the proportion of each strategy using the catches. And as you can see, the 5 most common strategy comprise about 77% of all the samples, while only 1/3 of the samples was sampled only once. Well, between the period of 1975 and 2022, we found a change in the CH at maturity. We found a marked and significant change in the proportion over the time of those individuals that mature at 2 years and the post-spawners, they increase, and those individuals that mature at 1 year, we saw a decrease. So, about the genetic structure and diversity, as you can see, we found a weak structure in which those locations from the mainstem conform an unique cluster, and the other tributary conform a separate and disparate cluster. In line with this, admixture shows us that the best K is equal to 5. As you can see, we confirm the information that these 5 locations from the mainstem share the same pattern of ancestry, and those tributaries show a different pattern. Then, with the FST values, we saw the same pattern, that those locations from the mainstem have the minimum values of differentiation, and the tributaries have lower values of differentiation. Then we evaluate the contemporary effective population size. And well, for this population, we can see that they experience a peak of about 800 and 500 individuals about 100 generations ago. However, there was a decrease reaching values of 200 to 100 individuals 13 generations ago. As we know, to ensure the survival of a population, we need an effective population size of about 100 individuals for a short time period, and for a long time period, we need about 1,000. So this river shows a concerning trend in the effective population size, considering the effective population size. So, to deal deeper in this weak structure that we found in Natamo River, it's important to mention that we have an absence of significant impact from straying hatchery rare salmon or escaped salmon in this system. So a possible explanation that we thought was straying from another rivers that are close to this system. And well, we have access to Teno River that is localized about 2,050 kilometers waterway. And well, as you can see, the circles that are in red correspond to Teno River, that is a massive system, and or Natamo correspond to the blue ones, and look like a tributary of Teno River. We analyzed the values of FST, and if we compare all the system with Natamo, we found values between 0.1 to 0.01. Then, if we compare Natamo with the mainstem and the upper rivers, we found values of FST from 0.08 to 0.01. And then, if we compare only those individuals from the mainstem, from the both system, we found values of FST from 0.02 to 0.01. So this can suggest that a possible explanation can be straying from neighboring rivers. And I said neighboring because between these two rivers, there is a lot of other systems that can be influenced Natamo. But we have access to Teno. So we found a generally wide life history with change in the CH at maturity, marked by an increase in individuals of 2 sea winter and a decrease of the 1 sea winter. We found a weak genetic structure and a decreasing trend in effective population size, possible the structure influenced by these neighboring rivers. So this kind of a study highlight the importance of protecting and continuously monitoring this kind of self-sustainable system. And well, thanks to Luke for the samples, to my project and my lab, and thank you.
That was super cool. I'm wondering if you were able to relate any characteristics about the life history traits with like river geomorphology, so like length of river or amount of spawning habitat or things like that. That would explain.
Yes, this is only an starting point because the project that we are working, we plan to compare a lot of phenotypes, different rivers, and we will include genotypes too. Important loci associated to life history traits like DGL, 3 that is associated to early maturation. So it's a big project. This is only the starting point. So the next one.
All right, let's start this. Hi, everyone. Thanks for making it to the last talk of the day. I know you're very ready to go to the poster session. I am Remy Gatins. I'm an assistant professor at Northeastern University. And today I'm going to be talking a little bit about some of the work I started as a postdoc in Katie Lauderhouse's lab that has kind of like spilled into, wow, there's a lot of bells. Has now kind of like followed me into my current position. So I'm going to be talking a little bit about the connectivity and kind of demographic history of a range expanding commercial species, black sea bass. So as many of you are very well aware, climate change is impacting our ecological processes. They're worldwide. We see more frequent and severe climate events every year, and particularly in marine systems, we have sea level rise that's impacting our coastlines, as well as ocean warming and ocean acidification, but mostly this is impacting species distributions. Either species are having to stay and adapt to their new environment, or they are starting to shift to more thermal optimum places. And here I just kind of put a plot of how it's been rising temperature, and it's just comparing kind of like the average of sea temperature. One of the main processes that has been impacted is species distributions, and this happens across very different taxa. And these are just kind of a few examples I was able to like Google really fast. But mostly marine species are particularly vulnerable to these, to ocean warming. And mostly because terrestrial species at least can kind of hide in these like thermal refugia, whereas marine organisms just kind of like sit in the soup and have to live with it. But these marine species are kind of like, they're at their boundary of their thermal optimum. They don't really have anywhere to go, so they can be more impacted. They also don't have these biogeographic barriers, so they tend to be kind of like shift really rapidly as well. And this is just kind of one of the plots from Pinsky et al., where they showed kind of that thermal refugia examples. Actually close to here, so one area that has been starting to gain more attention is the Gulf of Maine, and it is increasingly, it's heating much faster than most of the oceans worldwide. And this is mainly because the Gulf of Maine is actually kind of impacted by two main currents, which is the Gulf Current bringing up warm water, and there's this Labrador Current from the top. I feel like everything's very small, but trust me. And basically, as the Arctic and glaciers are starting to melt, it's changing salinity and changing density, and this water is being pushed away into deeper waters, allowing that Gulf Current to kind of come up more into the Gulf of Maine, so bringing these warmer waters. So we've started to see a lot more range expansions and new species, and I'm sure you've heard of cod moving up and lobster moving up, and so there's all sorts of different changes going on in the Gulf of Maine. One species that is particularly interesting to me is black sea bass. And black sea bass grow slowly. They grow up to about 2 feet, and they reproduce when they're about 1 to 3 years old. They are sequential hermaphrodites, so they change sex from female to male. And they spawn in coastal areas during the warmer months, and then they kind of move offshore during the colder months. And they feed on crabs or smaller shrimp and crustaceans. And they do have a very important commercial and recreational fishery. They are found throughout the Gulf of Mexico and all along the North Atlantic coast. And we believe they kind of like made their way up. I was very proud of that. They made their way up through the Gulf of, through the coastline, and they are currently being managed by 3 separate kind of management units, which is the Gulf of Mexico Stock Unit, Southern Atlantic, and Northern Atlantic. And these kind of, I guess, larger stocks or populations are actually driven, well, they were kind of designated because of genetics and life history traits, like differences between these. The genetics was mostly single mitochondrial marker differences. However, for this talk, I'm mainly going to be talking about this northern range as we're looking at this newly expanded range. So in the North Atlantic, that goes from like Cape Hatteras in North Carolina to kind of southern Cape Cod. And now we have that new expanded range in the Gulf of Maine. They have been kind of recorded or to be up into like mid-coast Maine in the last 10 years. So it's just basically a very recent population expansion. One fishery that's particularly afraid of him is the lobster fishery. They believe black sea bass are going to attack the young, and so there's a lot of studies right now looking at how they're going to interact together. And that's other PhD students in the department are actually looking at that. For my research questions, we were mainly kind of interested in like, well, what is the population structure of black sea bass across the northern and newly expanded range? And how has the demographic history of black sea bass changed through time? And so for this, we assembled the genome of black sea bass and sampled about 117 individuals across the northern and expanded range and did dd-RADseq to get at population structure in the area. And then to get the demographic history of black sea bass, we ran a PSMC analysis. It stands for pairwise sequential Markovian coalescent model, which we'll just leave it at PSMC. But basically, we're able to kind of, with one individual, we can see heterozygosity across the genome and understand how effective population size has changed through time. We also recently got some whole genome sequencing results from individuals across kind of range-wide. So we're going to start up in the north and then kind of expand. So without further ado, we do have a new chromosome level annotated genome that is really nice, and it was thanks to our funders in Revive and Restore, which we can now use as an awesome, incredible genomic resource. We mapped our RADseq to this genome, and overall, as you can probably tell, the structure, there's not a lot of significant structure up in the north, and so each bar represents an individual, and I've put them from north to south, so North Carolina in the south, but we do see that North Carolina, we do have some admixed individuals, and we mainly believe that these individuals are actually probably from the southern Atlantic region that might be moving up along as well. So what we used to think as Cape Hatteras in North Carolina being that boundary might actually be further north than we thought initially. So if we look at the PCA, it kind of shows something similar where the North Carolina individuals kind of cluster a little bit out. So we removed these North Carolina individuals to take a closer look at the northern range. And overall, there's not really a lot of structure. Here we are using all loci, 24,000 loci. And for the most part, it seems like there's pretty high connectivity. Now, this is not surprising, I guess. At first you get the data and you're like, okay. But it's not really surprising given that they've recently expanded, so maybe it's only been a couple of generations that have actually kind of like started reproducing up here, and that might not be enough to actually lead to a larger genetic kind of like genetic signature unless we were seeing like a strong genetic drift or something like that. However, when we look at the pairwise FST values, we do see that although it's weak, but there's somewhat significant differentiation between like Maryland, which is not surprising, which is kind of further south to most of the other populations. Massachusetts, I don't know if you guys can see my mouse. Okay. Massachusetts does seem to be somewhat different to the other populations. Interestingly, Maine, which is the furthest north, is not, and I don't know if that might have to do with kind of just the oceanographic conditions in the Gulf of Maine and Boston. There may be more self-recruitment in Boston, but yeah, that is one hypothesis. Now, looking at the demographic history of black sea bass, so using that one genome, we're able to kind of reconstruct how population size changed through time, and so on the y-axis, we have effective population size, and on the x-axis, we have like time before present. And so we see this interesting pattern, and the shaded area just shows the confidence intervals, but using that single genome and looking at the heterozygosity across the genome, it actually shows that there were like two main population bottlenecks. And this was, this was the PSMC analysis, and so you can think of these like population bottlenecks or the demographic history analyses being impacted by, there might be an oceanographic or a geological event like glaciation events that may have led to this, or it can also be impacted by population structure. However, interesting to me was there was this very, very low effective population size in the present. And if we go back to PopGen 101, population size and genetic diversity tend to be correlated. And so this individual of black sea bass was actually collected up in Rhode Island, which was the most successful sampling to date. They are everywhere. We caught 200 individuals in an hour. So it doesn't really seem that the population size is actually that low, but it may be because the genetic diversity is low. And so I hypothesize that these two bottlenecks may have been driven by these three population differences as they started to move up. Maybe there was a population bottleneck as they started to move up along the Atlantic. So it may just be that this one individual kind of shows that there's less diversity, just less genetic diversity up in the north. So we recently got back some range-wide whole genome sequences, and I was super excited to show you guys this until I got the results. And then, so now I'll leave it up to you. But we just got these range-wide kind of whole genome sequences for about 18 individuals range-wide, and so we mostly see kind of like an increase. We don't see those big population bottlenecks. I'm not sure. I at least assumed in the northern Atlantic, those individuals were from Virginia to maybe at least show the same pattern that we saw from that individual from Rhode Island, but we don't. If we break it down, the northern Atlantic just kind of seems like it increased and kind of plateaus, whereas the southern Atlantic has the highest effective population size. It also just kind of shows that effective population size increased. And the Gulf of Mexico, interestingly, did see a very tiny dip in the population size at some point. And so this, I mainly take away that the Southern Atlantic shows higher effective population sizes. It may be that the Southern Atlantic may actually be the true center of the range, and then the Gulf of Mexico and the Northern might actually be kind of like where they started distributing to. But I'm not really sure what happened to those two massive population bottlenecks that we see in Rhode Island. So take home, we end up with more questions than we do answers. But overall, we see high genetic connectivity across the northern Atlantic. It is not entirely clear what's driving those population bottlenecks up in the north. We still have to kind of look at other maybe geological events that might have happened. But the northern Atlantic and Gulf of Mexico populations do show lower population sizes, which may be lower genetic diversity. And black sea bass are a good system to study genomic impact and rapid range expansions. And so some of these things that we're looking at currently, we're looking in the northern range. We're actually quantifying the mutational load since it was such a rapid range shift. And if we have less individuals or less genetic diversity, we would expect to see higher mutational load in these range expansions. So we're about to send off for sequencing to get about 300 individuals, whole genome sequencing. And we did just get back a bunch of body condition metrics to measure fitness, which a student of mine will be presenting today. And we also look at range-wide kind of structure. So with that, I just want to kind of shout out to my students that are presenting today. So from here, go straight there. They're both presenting today, some on black sea bass and other of speciation and hybridization in angelfish. And I just want to thank everyone that has helped with this project in Katie's lab and all my students in my lab. And yeah, happy to take any questions if there's time. Yeah.
So I think I noticed this for the first genome, that was 30x covered,
It was
and the other genome,
15x.
you think that might play a part?
Yeah, good eye. I was wondering if someone would catch that. The other difference is that the genome was like long read sequencing PacBio sequences versus Illumina. I don't know if that, like the shorter sequences, has anything to do with it. But there was a paper that came out showing like different coverage and how that impacted this demographic history analysis. For the most part, yes, more coverage, but they said like more than 10x was actually pretty good. But more than 10x showed like the same patterns, just not as striking. But in this case, it, like, you don't even see it. So it could be, yes, like, I mean, we had approximately 15x coverage. It could be that, or it could just be the type of data we had of PacBio versus Illumina. Which is all the data at the end I was showing you, I'm comparing between each other, but I don't know if we can compare with the genome. But yeah.
So the first one was PacBio.
The first one was PacBio. So it might have to do with that, yes. Yeah. Any other questions? Otherwise, on the dot. I'll be around, but I'm happy to answer any questions after. Thank you.